I want to thank Asim wherever he is. He called me up and said he wanted to introduce me to a synthetic biology crowd. And I was kind of wondering what I had to do with that. But I put 2 slides at the end to try to make at least a tenuous connection of what I do to synthetic biology, so we'll see how that goes. So Dolly was cloned in 1997, and human yeast cells were described in 1998. And those 2 things are so intermingled in the public consciousness that people don't even distinguish them. And I think it's fair to say that the average person on the street can't even describe how cloning occurs or what an embryonic stem cell is because it's so hopelessly intermingled. But the reason those are connected in the scientific community was this idea that if you did somatic cell nuclear transfer a la Dolly, you could take a patient-specific stem cell or cell, turn it into an embryonic stem cell by going through cloning. And I've never been a big proponent of that because of economics. If you look at how you would have to source the oocytes and the inherent inefficiency of it, I just never saw it into the clinics because of the problems of cost effectiveness. And the real message of Dolly was not that you could clone sheep and not that you could do somatic cell nuclear transfer to make stem cell lines. The message was that the mammalian differentiated state is fully reversible and that there is naturally occurring transacting stuff in the oocyte that can mediate this reprogramming. And it was pretty clear even at that point that someday people would figure out what that transacting stuff is. And I think most biologists, including myself, thought it would be really complicated, that it would be hundreds of things, not 1 or 2 things. And my into this was about 6 years ago, I hired a very talented postdoc named Junying Yu. And when she showed up to my lab, I sat her down and we talked about this reprogramming problem. And we both liked it because we figured it was at least a 20-year problem and it would be something to kind of keep working at and kind of a nice hard goal off for the future. And it turned out to be much shorter than that. But she started 6 years ago, and oocytes are difficult to work with. It's very limited amount of material, and I'd done some nuclear transfer work in my thesis, and I knew how hard it was. So we decided at first to resurrect an older model from the 1970s. And this is before embryonic stem cells have been derived, but there are these things called embryonic carcinoma cells, which are the malignant counterpart to embryonic stem cells, basically. And in the mid-70s or early 70s, people figured out how to fuse cells together, and there's a whole cottage industry of fusing this cell with that cell. But one of the cell types, or the pairs of cell types that they fused together, are embryonic carcinoma cells with blood cells from a mouse spleen. And they found in that fusion product that the blood-specific genes were turned off, and that the SEC cell-specific genes stayed on, and that heterokaryon, it has various names, that fusion product had the developmental potential of the embryonic carcinoma cell. So this is actually evidence a couple decades before Dolly that there could actually be reprogramming events in mammals. And this had only been done in mouse at the time that we started this work. We decided as a first step to kind of resurrect this model with human material. And Thomas Waka several years ago made this knock-in cell line in human embryonic stem cells. And the green fluorescent protein and the drug-selectable marker are driven off an endogenous ESL-specific gene called OCT4, which you've heard about in Rick Young's talk. So basically, in the undifferentiated state, the green and the drug-resistant, and when they differentiate, the green turns off, the drug resistances go away. And because of this, we thought we could screen for reversions back to the undifferentiated state using the drug-resistible marker. So Igor Slutkin, a very good collaborator who studies hematopoiesis, took these cells, differentiated them to a particular blood type that he's good at making, a myeloid precursor cell. He could make large numbers of those. We confirmed that the GFP was turned off, that all the SL-specific genes were turned on, and they turned on all the myeloid genes. We then took that cell type, fused it to an undifferentiated embryonic stem cell, and examined the characteristics of that fusion product. And that fusion product turned the green back on. So it had at least OCT4 turned back on, turned off all the blood-specific markers, and turned on all the cell surface-specific markers of embryonic stem cells. And indeed, it looked, all intents and purposes, like an embryonic stem cell. So we saw that this is a starting point. We could compare the transcription profiles of these cell types and start looking at the differences to try and understand what transacting stuff made by the embryonic stem cell could mediate this reprogramming event. And this is before all these nice, cheap, high-throughput sequencers, but we had a nice collaboration at Vancouver with Mark Amara, and he did very deep SAGE analysis for us for both the blood cell and for the embryonic stem cell. And we simply digitally subtracted those 2 populations. And we came up with a list of 250 high-priority genes, which are either transcription factors, things that bind or modify chromatin, and RNA binding proteins. And Janine set about cloning all of them, all 250. And at the same time, she optimized the lentivirus vector to introduce these genes to study reprogramming. And she got about halfway through the list and started putting them into pools to see if we could mediate reprogramming. And when we started these experiments, we didn't really think it would work, or at least not fully. Our expectation was we'd get some partial reprogramming events, which kind of go in the right direction, and that over time we might build up enough knowledge to do it properly, but we figured that this would be a 20-year project or something. So this is the first list of genes, and the very first list of genes worked. So this is the first 14 out of 250. But of course it wasn't random. She basically went through the literature and picked out her favorite genes for which there's good evidence they're developmentally important. And it worked because, again, we were using these targeted knock-in cell lines. They reverted to expressing EGFP. They expressed all appropriate cell surface markers. They formed teratomas just like embryonic stem cells. And indeed, you couldn't distinguish them from embryonic stem cells. So at this point, we were very happy with ourselves. We're jumping up and down. However, this is right at the point where Yamanaka published his mouse reprogramming work. And if you think about it, although we're disappointed at the time, and we certainly are still, it was inevitable that the mouse system would win. So the rhythms of developmental biology for humans is 9 months, and for mouse it's 3. And those rhythms are pretty much recapitulated in vitro with human embryonic stem cells. I think I just turned off. With human embryonic stem cells. So the cycle time to do one of these reprogramming experiments with mouse material is about 2 weeks, give or take. Our cycle time was 2 to 3 months. So to go from those original 14 factors, which we had sometime early in 2006, to the final 4 that mattered, it took us 18 months to sort through all those things. And at that time, the mouse community raced far ahead. And we're still hoping that there's species-specific differences so that those factors Yamanaka identified would work for human. But it turned out they did work. But you had to change levels and timing, some other things. But it did ultimately work. And in our case, we came up with 2 that he had previously identified, OCT4 and SOX2, and then 2 others which he had not previously identified. And one point I'd like to make is that if you actually look at one of the reasons I thought this would fail to begin with is if you look at the combinatorics of even a set of 250 things and you say take them 10 at a time, that is an astronomically huge number. You can't sample the whole combinatorial space that you want to do. So we got lucky by having a lot of previous biological knowledge. So in some sets, that combinatorial space has been sampled twice independently because I didn't know it was on Yamanaka's list and vice versa. But it's not really independent because he packed his list with all his favorite developmental biology genes and they overlapped a whole lot. So that space has been sampled very, very incompletely. And most of what's happened since is people take our genes or his genes and combine them and try to get rid of them and add. But nobody's actually independently gone back to that space and said, are there other sets that can do this? So I think we should keep an open mind that there might be additional sets that can do this that are entirely independent of the ones that were previously identified. And I should make one comment. So in our case, this is a subtraction experiment, and this is colonies of these iPS cells formed when we subtract each of the factors. This is all 4 together. If you take OCT4 out, you completely wipe out reprogramming. If you take out SOX2, you completely wipe out reprogramming. If you take out NANOG, you get a dramatic reduction, but it's, at least for this early scoring, it's not absolutely required. In Lin28, we got a 2 to 3-fold difference, but it looked like it was expendable in these early experiments. And when we took several clones, expanded it into the teratomas and examined them, we found out indeed that from 2 different cell lines, one clone actually lacked Lin28. So we knew really early on that Lin28 was not absolutely required. NANOG, at least for the early steps, is not required. We don't know about subsequent steps or not. And I'll skip a lot of this early work just to say that they have all the properties of embryonic stem cells that we originally defined human embryonic stem cells as, except being derived from the embryo, including all the markers of embryonic stem cells, normal karyotype, telomerase, and they form wonderful teratomas which are indistinguishable from embryonic stem cells. Now the problem, of course, with this early work is that we and others were using retrovirus or lentivirus vectors to introduce them. In our case in particular, the lentiviruses were very leaky, so the transgenes stayed around a little bit. They weren't completely silenced. And there's a very reasonable fear of integration into genes that would affect the behaviors of the subsequent cells. So it was very desirable to have a way of reprogramming these cells in a way that you don't actually have to integrate in the genome that's without the transgenes and without the vector sequences. So Bill Sugden's here on campus, and he's kind of the expert of these EBV vectors. And probably 10 years ago now, we started seeing how well these would work with embryonic stem cells, because back then they were very difficult to transfect. And what we found was, at the time, kind of disappointing. This is transfection. So for these EBV vectors, you need a transacting factor called EBNA1 and an origin of replication called OREP, and that's sufficient, at least in a subset of cells, to make this episomally stable. And what we found in an EBNA1 expressing human embryonic stem cell line, if you have the OREP versus not having OREP, there's clearly an advantage in the transfection efficiency of the OREP, but both crashed within about 2 weeks. And in a typical somatic cell, the loss early on is less, and although it doesn't look like it in this graph, you usually get like 1, 2, 3% of stables out here. So early on we thought, well, if you have to introduce something for maybe 2 weeks, there's a little advantage of it, but we didn't use the vectors very much because they're clearly not very stable. But coming back to the reprogramming, we thought, well, maybe this is what we actually want, is that in the differentiated cell, you're getting pretty good expression for about 3 weeks. And in embryonic stem cells, they're lost at a pretty high frequency. So if you could mediate reprogram because they're around long enough, and yet they tend to be lost in the embryonic stem cells, you might just have a really good way of introducing the gene and then have it go by without much effort. And that turned out ultimately the case, but it took us a long time to get it to work. In particular, because the efficiencies of the episomal vector are relatively poor, we knew we had to increase our reprogramming efficiencies a lot from our original work. And Jining spent a lot of time linking the genes together, and by using the appropriate combinations of irises and things, you get about a 10-fold increase over what we had in the original paper, which was a step in the right direction, but not enough. We then looked at the factors that Yamanaka had described, and we found by combining his genes with our genes and linking them just the right way, we got another 10-fold increase in reprogramming efficiencies. So Jining could very consistently get about a 1% reprogramming efficiency, both in foreskin fibroblasts and in adult cells, which is actually, in this field, a very good reprogramming efficiency. We then decided that this was high enough that we could try the episomal vectors. She used a combination that linked Yamanaka's factors with our factors. And in every combination, it failed. And what she noticed is in the early cultures, there was a huge amount of cellular death. And although c-Myc is relatively innocuous in mouse embryonic stem cells, in our system, we believe that was associated with induction of apoptosis. And we decided to add SV40 large T antigen because it kind of counteracts the negative effects of c-Myc. It binds p53 and other things to reduce apoptosis. And after empirically putting these genes together in hundreds of different vectors, we had 3 combinations that worked, all with these particular set of genes. So this is not satisfying from the point of view of it being highly empirical, but it does actually allow the introduction of these cells with this episomal vector and reprogramming. And once we subcloned them, the vector was clearly gone. The RNA from the transgenes is gone, both by PCR and by southern blots. The vectors are entirely gone. Similar to human ES cells, they have an identical morphology. Most of them had normal karyotypes, not all. We only looked at a couple promoters, but the promoter region for OCT4 and NANOG were both demethylated during reprogramming. And again, the specific cell surface markers of embryonic stem cells were expressed. Now, what impressed me about this is that when we did cluster analysis and looked at the various clones we derived that were reprogrammed, we did the analysis at the same time with all the original human embryonic stem cells we had, which were 5 of them. And this happened in every case of reprogramming, not just the non-integrated ones, but also the integrated ones. The reprogramming clones always clustered together in a very tight little cluster. And the amount of variation between the different human embryonic stem cells, they're always clustered in the middle of the embryonic stem cells, but the amount of variation about the human embryonic stem cells is always greater than the iPS cells, which surprised us because at least going into this, there's a lot of variation when you do somatic cell nuclear transfer allodoly, and we thought that that would be comparable in this, but at least in our hands, they're tremendously similar with each other when you look at the RNAs within the embryonic stem cell itself. So why does all this matter, and how's my time doing? How about you? Okay. Basically, I wrote something like this 10 years ago for the S cells, and I just noticed I'm getting cropped to the RNA. The reason these cells are important are the challenges for using these in transplantation therapy, which again I've been always a little bit skeptical of, is first making the cell type of interest. Well, developmental biologists are getting pretty good at making a lot of cells, and I would guess within the next 5 to 10 years we'll be able to make essentially all the clinically relevant cells in the body in quantity. But it's all the other things which I think are really hard, and why I think that transplantation cell based on these cells is going to be a very rare event and maybe not even occur in the next 5 to 10 years. Safety concerns is a really big one, and this is true with any cell type that's been cultured for an extensive period of time in tissue culture. If you think about type 1 diabetes, that's a disease where transplantation really should work. There's already a transplantation procedure based on cadaver tissue, and there's nasty immunosuppressive therapies, but it does work. Type 1 diabetes is a nasty disease, but a child will live a long, productive life with that disease. If you get pancreatic cancer, that kills you really fast. And if you accumulate enough mutations in tissue culture, you have at least the possibility of transforming that to a cancer. And it's really hard to figure out how you're going to screen for that ahead of time if it's like a 1 out of a billion cell event in a population, because you can look at the global population, but it's hard to look at these rare populations. So I think those safety concerns, especially for things for like diabetes where there's an alternative decent therapy already, are going to be very, very challenging. Immune rejection, iPS cells, kind of solves, but if you think of important diseases like diabetes, that's an autoimmune response, and making genetically identical cells doesn't solve that one either. But these are the two that I've always thought that would be kind of a deal stopper for most of the diseases you see in the press, like Alzheimer's, for example, forget it, and even something like Parkinson's, where it's a very specific defect. You have to prevent the process that killed the cells in the first place, and at least for Parkinson's, that's not well understood. And integration back in the body in a physiologically useful form will be very challenging. So my personal feeling is that for most diseases, this is really, really hard. But at some kind of extremes, if you think about cells that don't need high integration into the body, for example, if you think about the blood system, whether thinking about transfusions or transplantations, there's already existing therapies for that. It's all well worked out. And I think in the blood system, especially for leukemias, where if the patient doesn't actually have a match, they're willing to tolerate a pretty fair amount of risk. And if you can use iPS cells to create that match and get a graft blinded stem cells, that's one that should work. But that's kind of at the extreme where the amount of physical integration is kind of small. When you take to the central nervous system, I think that'll be challenging. So I think therapies based on these cells will be few and far between. But where the true value is that they give us access to the building blocks of the human body for the very first time. And I think this will be used in ways that have nothing to do with transplantation therapies, but there's species-specific differences between you and a mouse, and where those are clinically significant, these will be extremely valuable. And the human heart is certainly one of those examples, and this is just an example of looking at the channel that's involved in prolonged QT polarization, which, if you know what that is, it's stopped a lot of drugs getting to market. So I got 2 more slides. I think I'm on time. I decided to say something about synthetic biology, and it's a real stretch. If you think about Ian Wilmot and my work with embryonic stem cells, we haven't made a very large economic impact on the world. Unless you're a bioethicist. We've been really good at having medical schools hire a lot of bioethicists, and we've been really good at having their mortgages paid for the last 10 years. So the question is, with embryonic stem cells kind of drifting away and iPS cells coming to the forefront, do they have to worry about paying their mortgages? And I believe that now that the real challenges are just occurring because iPS cells, which is kind of contrary to the way everybody's looking at it. Prior to deriving human embryonic stem cells, I thought there were 3 ethical problems. One is you kill an embryo, and that's going to piss off a lot of people. But there's also the use of the technology. And mouse embryonic stem cells are routinely used for making transgenic mice. And there's also reason to believe that they're good at cloning, and they are for technical reasons. And back then, Dolly had already been cloned, so I figured the cloning argument didn't matter anymore. And the transgenic one, the way you make transgenic mice, it takes basically 2 generations. You got to make a chimera, it's got to go to term, it's got to, so it's a long time in human time, and we're too lazy. It's not going to happen anytime soon. However, iPS cells are different. And so this paper, if you're not in the field, you probably didn't notice, but there's at least 3 groups now that have made entire mice out of iPS cells. So this means you can take a fibroblast, change it with like 4 genes, and without going through the germline or noocyte or anything, make an entire mouse. That sounds something like synthetic biology, I don't know. And the way this is done is you take a few cells that normally make the placenta, you intermingle them together, the placental cells go and make the placenta, and the iPS cells make the mouse. And it's born, and the placenta cells go away, and you have this entire mouse in 1 generation from a tissue culture cell line that came from a skin cell. So I don't think that this is where the ethical problem is with humans. I don't think people are going to do this right now. But I can now see a series of stepping stones where we're going to get into ethical areas that people weren't going to get to with embryonic stem cells. So let me finish on this slide. About the late 90s, and I can't read this, it's kind of blurry, but Ralph Brinster figured out how to transfer spermatogonia between mice. It's real efficient. He's really good at it. A lot of groups have done this and repeated it. And people have thought that, well, you can use this for cancer patients, because if you take the spermatogonia stem cells out of the cancer patient, treat them with nasty chemicals that protects their germline when they're cured from leukemia or whatever, you can put the germ cells back in. And people are showing this in mouse models, for example. So it's already talked about as a potential therapy. About a week after we derived human iPS cells, a guy from another country, and a scientist, but not in my field, emailed me and said he, as a kid, had a medulloblastoma and had been very aggressively treated with chemotherapy, and it was completely cured. But now he's a young adult, and he married recently. And he said, could this new discovery with iPS cells cure my infertility? And I basically wrote back and said, well, not right now, but yes, it's actually very possible. Embryonic stem cells and germline stem cells are very closely related developmentally. A bunch of people are studying germ cell development, and I think the ability to make those cells are going to happen in, the next 5 years or so. And I think if you're that individual, a number of societies would say, well, you're only taking his own cells and correcting something to him, and they'll allow him to do that. But the add-on question, of course, is, well, there's this genome project now, and I had this nasty medulloblastoma, and there's these 3 mutations, which if I corrected them, my child would not have that. As long as you have them out, would you please correct that? And I think a number of societies would say, yeah, that's kind of a reasonable thing, because why should his kid go through the medulloblastoma again? And because of that, I can now see these incremental stepping stones, which I didn't see before. So my confidence is that those mortgages with the bioethicists are going to be paid off really well for quite some time. I just want to thank a lot of people in my lab contributed, but Jinjing Yu was the driving force behind this. She worked on this 5 years in my lab, and excuse me, the reprogramming papers are entirely her work. Thank you.